Welcome back to another BGCSE paper review. Today we're reviewing another biology paper, a paper 2, and this is from the year 2019. Now always remember to write your school number, your candidate number, your surname, and your initials. Also, very importantly, you need to read through the instructions carefully, and you should not open the booklet until you are told to do so by the examiner. Now let's go into our first question. The first question reads that the diagram shows the national fish of the Bahamas. The binomial name for the national fish is the Macaria nigricans, which is the blue marlin. Now the question now asks us to, which is part A, it said, what genus does this fish belong to? Now always remember that the scientific name or the binomial name contains two names, the genus and the species. So the genus is the first part of the name, start with an uppercase letter. And so let's write in our genus right here, which is the Macaria. And so it is Macaria as the name for the blue marlin, which is a genus. Now, part 2 of A state what does the term binomial means. So binomial, as the word bi means two, and nomial mean a naming system. So this is a two name system. So this is a two name system involving the genus and the species. The genus and the species. All right, so it's very importantly for you to know that. So it must include the genus and the species. All right, so it's a two-name system, binomial. The next part of the questions asks us now to state the importance of using the binomial name. The binomial name is the same thing as a scientific name, and the scientific name is an universal name. And so therefore, it used to prevent confusion. So anywhere you go in the world, this name will be the same. All right, it's different from the common name, which can vary. So this one is to prevent confusion. So to prevent confusion. All right, so to prevent confusion. All right, and the reason why prevent confusion, because the name is used internationally. Okay, so you can make a note of that, that the binomial name or the scientific name is used internationally. All right, so let's go to the next part of the question here. And this part is a complete classification of this organism by filling in the table below. And so here we have the kingdom, which is the largest group that this organism belongs to, which is the Animalia, so it's an animal. Now it asks us for the phylum, and the phylum, since the fish here has a backbone, it belongs to the phylum Chordata. Okay, so it is Chordata. And so the class of fish, the order is the Performs, and then we could go into the family, which is the Istophoridae. And then the genus, as we have in the scientific name, which is the Macaria. All right, and the species here will be the nigrican. All right, so that will be the species. All right, so now we have the scientific name and the whole thing filled out. All right, so we could jump to the next question. Or the part of the question, this is part C of the same question. And this part reads, it said that select two organisms from the list given to complete the food chain below. And so here we have plankton being shown starting there food chain. So these will be phytoplankton. So you can make a note of this. Matter of fact, let me write this in for you. So these are planktons, and so these are called phytoplanktons. Remember, we also have zooplankton. Zooplankton those are animal-like. So phytoplanktons, they are plant-like, and so they are producers. Okay, so phytoplankton. All right, and so what will eat phytoplankton will be the shrimp. So the shrimp could be placed right here. Okay, and then what will eat the shrimp from the list up here, the tuna will eat the shrimp. The tuna could eat the shrimp. And then the blue marlin will eat the tuna. Okay, so that completes the food chain. And so let's go to the next part of the question. It's the last part of this question. It said, identify the trophic level of the following organisms in the food chain. And so it's a trophic level. Remember what trophic level is. The trophic level is each feeding level of the food chain. So the plankton belongs to the first trophic level. So you could write that as the first trophic level. Okay, first trophic level. All right, ST. All right, so that's the first trophic level. And just to make some additional notes here, that the phytoplankton is the producer. Okay, so this is just some extra information here. And then the blue marlin, you could count 1, 2, 3, and 4. So the blue marlin is the fourth trophic level. So this is the fourth trophic level. That's the fourth trophic level. And just to put some additional information here again, that is also occupying the tertiary consumer. Okay, so this is just some additional information here for you. Tertiary consumer. But it is the fourth trophic level based on what the question is asking us. First trophic level, fourth trophic level would be your answers. But I'm only putting these extra just for additional information. All right. Now, one thing to point out in food chains do not show omnivores, just to point that out, because omnivores eat both plants and animals, and a food chain only shows one feeding relationship, which means only one food source, okay? One food source. So just to point that out. All right, so you can make notes of those information. So let's go to question number 2. And in question number 2, you said the diagram shows structure A and an associated blood vessel in the human respiratory system. So we have structure A, which is this here. We're going to figure out what that is. As a matter of fact, we can actually label it. All right, so we could go ahead and label what that is. And so here we have this structure being an alveolus. So this is an alveolus. All right. And so blood, B will be our capillary. So B is a capillary. All right. So B is a capillary. All right. And capillaries surround the alveoli. And C will be the red blood cell. All right. And so let's see what they're asking us in this question. And so for part A, it said on the diagram, draw an arrow to show the direction of the movement of carbon dioxide between blood vessel and the structure A. So looking for carbon dioxide. Now remember that carbon dioxide will leave the blood, go into the alveoli, and go out of the body by the process of exhalation. So our arrow right here representing carbon dioxide should leave from these blood cells inside of the capillary and go into the alveolus. Okay, so very important to note that. All right, now let's see what the other part of the question is asking us. As a matter of fact, let's label this as CO2, as carbon dioxide. So it's good to also label. So this is carbon dioxide, which is CO2. Okay, so that is carbon dioxide leaving the blood capillaries and go into the alveolus. All right, so next part of the question here now. Reads is a name gas D and state the name of the process by which it passes into cell E. So I want to label name this gas D, and the arrow is coming in. I'm not showing so properly. Let me just draw that for you real quick. The arrow is showing you coming in this way. Okay, so the arrow is going in. All right, so this now is the gas that leaves from the alveolus into the capillary is oxygen. So this is going to be oxygen. It's going to oxygenate the blood. So carbon dioxide leaves the blood and oxygen will enter. So this is oxygen. Okay. And I said name the process by which the gas will enter. And so the gas is leaving from the alveolus into the blood capillary by a process of diffusion. So the gas will diffuse into the blood capillary. So this is diffusion. All right, great. Now the next part of the question here said name the structure labeled A. We already did that. So structure A was the alveolus. So you could just put it back here. Is the alveolus. All right, we already go ahead and label that. So alveolus. All right. So that's the alveolus. All right, excellent. Now, part C now, it said identify the type of blood vessel labeled B and state one structural characteristic of this blood vessel. So the blood vessels that surround the alveolus, they are capillaries, okay? So capillary is our answer. We already labeled that as well. So here is capillary. All right, so that's a capillary. And one structural characteristic of a capillary. Capillary is characterized by its very thin walls. And to be specific, the walls are thus 1 cell thick. So the capillary is just 1 cell thick. Those put that in real quick. They are 1 cell thick. That's how thin they are. They are thick as 1 cell, very thin. All right, so great, 1 cell thick. Or I can say very thin. All right, so part D of this question now reads that it says state the term that describes the composition of the blood entering B and name the part of the heart that this blood came from. So the blood is coming from the heart, and we know that the blood that comes from the heart, it is deoxygenated blood. It comes to the lungs, so it can be oxygenated. So it's coming from the heart, and so this is termed as deoxygenated blood. Deoxygenated. All right, so deoxygenated blood. All right, so this is deoxygenated blood. All right, so that's the term used to describe the blood. And then now it asks us now, it's a name the part of the heart that this blood came from. And so it came, if you think about the heart, just remember, if you can remember Avlov B that I talked about in the previous paper, then you know that this will come directly from the right ventricle, okay? And also remember, a matter of fact, you need to remember that blood leaves the heart from the ventricle, okay? So this is the right ventricle, okay? The right ventricle. So that's where the deoxygenated blood came from and enter the pulmonary artery and will go into the lungs. All right, so let's look at part C. It said name 2 respiratory diseases caused by smoking. And so smoking could cause a number of things. So let's look at one of them could be bronchitis. And so let's write that in right here real quick. So bronchitis. All right. So bronchitis is one. And the other one is emphysema. It is EMPHYSEMA. Emphysema. I miss a letter, right? Y. PHY. Right. Awesome. And then just to make a note, and this is not necessarily will happen, but this could be another possible. You could also have lung cancer, right? We're not saying that smoking will always give a lung cancer. I'm saying it's a possibility here that you can have lung cancer as well. All right, so those are possible respiratory diseases that you can have by simple smoking. Let's go to our next question, and this is question number 3. And question number 3 reads that macronutrients are nutrients that are required in relatively large quantities in the diet. So the word macro means large, And so, I mean large nutrients that are required in the body or required in very large amount. And so, it says state the elements found in all macronutrients. And so, the elements that you can find in all macronutrients will be CHO, which is carbon, hydrogen, and oxygen. So let me just put C here, and C present for carbon. So just in case you want to know the name, and it says state the elements, you prefer to write the names out here. So carbon, or you can put the name and then put the symbol in brackets. So carbon, hydrogen, and this is symbolized by H. I could put H at the front of it. Okay, so H there. All right. So H is for hydrogen. And we also have oxygen. So oxygen symbolized by O. So oxygen. So these are the elements present in all macronutrients. And the macronutrients, just to point them out, so just in case you need to know them, we're talking about proteins, carbohydrates, and lipids. Okay, so those are the macronutrients we're talking about. Proteins, carbohydrates, and lipids. All right. Of course, protein will be different from the others by containing nitrogen. So what separates protein from the list is proteins contain nitrogen. But lipids and carbohydrates only contain C, H, and O. All right, part B is the proteins are example of macronutrients. It's a name the 4 elements found in all proteins. Okay, we'll just talk about that. So the elements found in all proteins here will be carbon. So let's just put them. I'm not going to put the symbol here now since we already have them up top. So we have carbon, we have hydrogen, okay, and we have oxygen. And the last one here will be nitrogen. Nitrogen is symbolized by uppercase N. Okay, so nitrogen. So nitrogen is what separates protein from the other macronutrients. Okay, nitrogen is present in proteins. Okay, so for part 2 here now, it says state the building blocks of a protein molecule. And proteins are made up of what they call amino acids. Okay, and so this is a good question as well, because it could also ask what are the monomers formed when they break down proteins. So when proteins are digested or hydrolyzed, we get amino acids as the end product. Okay, just to point that out. Part C is a diagram shows 2 types of carbohydrates. A carbohydrate again is another example of a macromolecule. And just by looking at this, we can tell what they are. And so these are single units. W represents single units, and single units will be single sugar or simple sugar, otherwise called monosaccharide. Okay, so let's put that in real quick. All right, so monosaccharide. All right, and then the X will be our polysaccharide. Okay, so polysaccharide. All right, and so those will be our carbohydrates listed there. So monosaccharide, polysaccharide, okay? Just to make a note of that. So please make sure you know them. All right, so polysaccharide, monosaccharide, okay? All right, good. All right, so let's now jump on now to the question. Said identify a type of carbohydrate labeled W and X, which we did. Okay, so again, W is our monosaccharide. Let's put that in real fast. Okay. All right, and then we could go into an example of monosaccharide. Remember, those are the simple sugars. So monosaccharides are simple sugars. Three examples that you must know. I'm going to put all 3, but the 3 examples of monosaccharides that you must know include glucose, galactose, and fructose. Okay, and just to point out this as again that sugars end with OSE, So these are all sugars. So galactose, glucose, fructose. And X, again, because it contains many single units, we call that polysaccharide. So this is a polysaccharide. The polysaccharide. And examples of polysaccharide, three examples that you also need to know. One common one in plant that everybody should know is starch. So starch. Plants also contain another polysaccharide called cellulose. And in animals, we have glycogen. Okay, so and glycogen. Let me get back into this. And glycogen. Okay, so those are the polysaccharides that you need to know. We also have disaccharides. I'm just going to quickly mention disaccharides. And disaccharides include maltose, sucrose, and lactose. Okay, so those are the three disaccharides that you need to know. Okay, at least three of each. Now, part two of this is carbohydrates are the key energy sources in the body. It's a name the carbohydrate stored in the liver, and in our liver we stored glycogen. So glycogen is stored inside of the liver. Okay, so glycogen is a stored form of carbohydrates in animals, as a matter of fact. All right, plants will store their carbohydrates in the form of starch. All right, now let's look at part D of this question. It said, today many processed food undergo food preservation. It says, state the meaning of the term preservation, what it means to preserve. And so preservation is a process, by the way. So preservation is a process. It's a process or a method. You can even use it as a method. It's a process or a method used to prevent or to delay. To prevent or delay. I can put delay as well. To prevent or delay. Let me put delay right there. To prevent or delay spoilage. So delay spoilage or damage. Okay, so spoilage or damage. All right. So once it prevents any delay of spoilage or damage to a substance or item. Okay, to a substance or item. All right. All right. So that's what preservation is, to preserve something. Okay. So you prevent the delaying of spoilage or damage to that thing. So in other words, you increase its current state or to maintain its current state for a long period of time. All right, so the next part of this question is, name a specific chemical used to preserve meat and fish and suggest a possible health risk of this preservative. And so the chemical used in preserving meat and fish, a simple chemical that we see every day, and even in our kitchen we have this, and it is salt. So salt can be used to preserve fish. A common fish that is preserved using salt is the codfish. All right? And so what we're going to do here now is to list the health risk. I am also going to mention another chemical, but it's illegal, but I'm still going to mention it. And remember, the particular salt is used, I must write that real quick, is sodium hydroxide, okay? It's sodium chloride. I said hydroxide, my apologies. So sodium chloride is used as the salt, okay? All right, and the health risk in using sodium chloride, there are so many if you intake salt. But the common ones you need to think about is high blood pressure. You may have high blood pressure, okay? So high blood pressure could be a result of this. Also, it could also cause other heart diseases, even cause stroke if you take in too much salt. So stroke and heart disease, okay? So heart disease could also be another one. But high blood pressure is the most common, right? Another chemical I want to mention here for you, right? It is illegal. However, it is illegal, but I'm going to mention it and give you the health risk here. Again, they will not typically ask you for this, but it's called formalin, okay? It is called formalin. And formalin will lead to cancer. It is cancerous. All right, so it can lead to cancer. Again, it is an illegal substance. All right, number 4 is a complete table to show the differences between asexual and sexual reproduction. All right, so we're going to talk about here, they ask us to list the number of parents for each of these. And asexual is 1 parent, while sexual involves 2 parents. All right, and so it's a genetic makeup of the offspring in relation to parent. And so the offspring compared to parent, it will be the same genetical makeup for asexual reproduction. Okay, but for sexual reproduction, they will be different. In fact, there'll be variation. So they will be different from the parent. Now, example of organisms that use this type of reproduction. There are many organisms that use asexual. So I'm going to list a few of them. So just in case, fern is a good one. So we have fern. We have potato. Potato is also a common one that we'll see on exam as well. You may see hydra. Hydra is another organism that will undergo this type of reproduction. Bacteria is also another one. Let me put it here at the bottom here. So we also have bacteria. So just follow the order which I'm doing is. Okay, we have bacteria, and another common one that we all use is yeast. Okay, just to name a few. So fern, potato, hydra, bacteria, yeast. Those are common organisms or things that you need to know that reproduce asexually. For sexual reproduction, we should also know that animals, generally animals such as ourselves, so human beings, also definitely will reproduce by this means, a human being. Okay. All right. What else? You can name a number of things, like goat, for example. Okay. Pig. Flower is the next one. Flowering plants, right? So a flower will definitely reproduce by this. So a flowering plant like a rose plant and so on, a citrus plant. So flowering plants tend to reproduce sexually as well because we have pollen and we have ovule that will fuse. Okay, so let's move on to part B. It's a state 2 advantages of asexual reproduction in plants. So what are 2 advantages of asexual reproduction in plants? Now, what I will always tell you is that whatever lists for advantage can also be a disadvantage. The difference is how you state these things, right? And so let's say, remember now that asexual is a fast process. It produces the same type of offspring as compared to the mother. Okay, so the offspring will be just the same as the parent. Okay, and okay, so generally same as parent. Now, the thing is this now. Since it's fast and short, we can produce a lot of offspring. Okay, a lot of offspring. And these offspring can be a lot in a very short time. So if it is desired, okay, so can produce a lot of offspring in a short time. In a short time. If desired. I'm going to put it right there, if desired, because remember again, if you do not desire a lot of offspring, then that becomes a disadvantage, right? And especially for invasive species and so on, then it could become a disadvantage. So again, producing a lot of offspring in a short time, that is if it is desired. Another one you could put here is that the traits or the characteristic can be maintained. So the quality of the parent can be maintained. The quality, because remember there is no change, right? So the quality of the parent or the plant, the quality of the plant, because you say this is in plant, so the quality of the plant can be maintained. And again, that is if you determine the trait of that plant to be quality. Let's say a sweet fruit, and you want to maintain the sweet fruit, the best way to reproduce that is asexually. If you reproduce the plant sexually, then the quality of the fruit can change because there'll be variation, right? So the quality of the plant can be maintained. Okay, can be maintained. I'll put that right here. Right, can be maintained. All right, let me just rewrite that. Can be maintained. All right, excellent. Another possible one also here is that there's no energy needed, no energy required, and let's write that one, or no energy or effort. I'm gonna put energy or effort. So no energy or effort is needed to find a partner or to find a suitable mate or a suitable plant to pollinate. Okay, so no energy or effort is needed to find a suitable plant for pollination, right? Or for mating or reproduction. So suitable plant for pollination or reproduction. Because remember now, doing it asexually, there's no need for any pollination at all, right? All right, so there's no need to look for, okay, which plant you want to pollinate this plant and so on. You don't need to look for that. If you deem that plant to be quality, it just reproduces asexually by cutting, by cutting, air layering, whatever the method you want to use. And so you could get that plant, even by root system, okay? All right, so great, that is covered. Now part C, it said name the type of cell of which ovum and sperm are examples and state the type of cell division that they are involved in. So they are definitely examples of gametes, or otherwise called sex cells. So they are examples of gametes. So gametes or sex cells. Okay, so gametes or sex cells. All right. And so it asks us what type of cell division they're involved in. And so the cell division, it'll put at the bottom right here. This will be meiosis, okay? Because meiosis produces gametes, which means half the number of chromosomes. All right? And so question number 5 now reads that this question is based on the process of photosynthesis in green plants. It states that option A state, complete the word equation for photosynthesis. What is the word equation? So first they put carbon dioxide. So what comes next? And it said word equation, not the chemical equation, but the word. And so right here, carbon dioxide will react with water. So this is going to be water. Remember, the plant needs carbon dioxide and water. So this is going to be water here. It will produce glucose and oxygen. So oxygen will be a gas given off from the plant or plant-like organism that photosynthesize. Here we have light plus something. So light will be going into the chlorophyll. So there's going to be chlorophyll here. Okay, and remember the purpose for light going into the chlorophyll is to kick out those electrons, and those electrons will now use to split water molecules. Okay, so that will be now our answers. All right, so part B now, we said a student used a potted plant to find out if light is needed for photosynthesis. The diagram shows how she set up her experiment. Now, I want to notice something here. This is a potted plant. A leaf S is covered with a dark paper, and that the leaves were exposed, right? So just to notice that on the plant. All right, so the plant is treated like that. All right, so part 1 now says state what the student should do next with the plant before carrying out any test on the leaf. Now, remember what she's testing for is the testing if the plant test the effect of light on photosynthesis, or if light is needed for photosynthesis. So how are you going to test for light for photosynthesis? Test if starch is produced, right? So if you're going to test for starch you're going to produce, there are 3 things that you're going to do to carry out this experiment. There are 3 things. And so the 3 things will include, let me just write them up here quickly and then I put them on the bottom. To carry out an experiment like this, you must always start with 1 is to de-starch. So I'm going to give it in short right here. So you want to de-starch the plant. After de-starch the plant, the next thing you want to do is to place the plant in light. So the purpose for that is to restarch. So you want to de-starch, then restarch. Okay. All right. So let's put that one in real quickly. Okay, so de-starch and re-starch. Okay, so that's what you're going to do right there. All right, I change this right here. Okay, all right. So you're going to de-starch, you're going to re-starch. All right, de-starch, re-starch, de-starch, and re-starch. Those are the things you're going to do. And the next thing you want to do is the third thing is a test for starch. Okay, so it's all about starch. Test for starch. Okay, test for starch. All right, so first thing, you de-starch, you re-starch, then test for starch. Those are the three things you do to prove if the plant is photosynthesizing or if a factor is needed for photosynthesis, right? Because you're testing for starch, you take out the starch first, then you re-starch the plant and you test for the starch. All right, and what you're going to do, how you're going to re-starch is put it under the condition to see if it will re-starch, a matter of fact. If it does not re-starch, then that factor is not needed for photosynthesis. All right, so let's go to answer this question now specifically. It said, state what the student should do next with the plant before carrying out any test on the plant itself or on the leaf. So the first thing she must do, or the thing that she must do here, is to de-starch the plant, okay? So so the first thing she's going to do is to de-starch, okay? So one here is to de-starch. Remember, we're going to de-starch the plant. Okay, so de-starch the plant. That's the first thing. And how you de-starch a plant, you're going to put it in a dark place. Okay, so de-starch a plant by placing the plant in a dark area. Now, why you do this? Because if a plant is in a dark area, the plant will no longer photosynthesize, and instead what will happen, the plant will use up the stored carbohydrate to make energy by respiration. So photosynthesis will decrease and respiration will increase. So the starch that was stored will eventually be used up, okay, in the leaf particularly, okay, not in the roots and so on, but in the leaves. Now, the next thing she needs to do before she starts doing a test is to re-starch it. So she's going to re-starch, to put it right here now. So the next thing she's going to do is to re-starch a plant, okay? So re-starch the plant or the leaf. You might want to say the leaf or the plant, really doesn't matter. So re-starch a plant by placing the plant in light. And remember, she's looking for light, okay? So the factor that she's testing here is light. So re-starch a plant by placing the plant in light. All right, and that will now allow the plant to now photosynthesize if light is needed for photosynthesis. And once photosynthesis takes place, then starch should produce. Okay, and remember now the last thing she want to do is a test for starch. And I'm going to tell you right here, but this again is not a part of your answer. So let me put it right here. You test for starch by doing a number of things, right? Let me just check if the question is actually asking that and see. So name the product of photosynthesis, name the reagent used, outline below. No, okay. All right, so you're not asking for the step, but I'll give you the step, how you test for starch at the bottom here. Also, you could see my lesson on that. So let's just answer the next part of the question, right? And I will show you how to test for starch in a very short way. Okay, so name the products of photosynthesis that could be tested in this investigation. And this product that we're going to test here for is starch, because starch is an indication that photosynthesis would have occurred, okay? Is it name the reagent used to test for this product? And what you use to test for starch is iodine. So you use iodine right there. Number 4, it said the outline below represents leaf S, not drawn to scale. It's a complete diagram and show the result of the student investigation. Label your diagram clearly. So let's do some drawing here now. We're going to outline what is happening here. And so remember the leaf. Let me just go back quickly and show you how the leaf appears. Remember the leaf was covered with a black object or a black piece of paper or sheet or whatever it is, but something that is dark that prevents light from going in this section of the leaf. So on this section of the leaf, we're going to make a marking there and we're going to label this. So first, let me just put some marking right here. All right, let me use black for this to make our markings. All right, matter of fact, let me use purple. All right, I just want you to see this clearly. So one marking will be here to represent one edge of the paper or the dark object, and another piece will be here, right? So that part was covered with the black object or the dark object to prevent light from going in. Now, let's kind of label this, right? What will happen here? So let's go from top again. Let me label this portion right here. So these two areas that I'm going to point out right here, these two areas, they were left exposed to light. Okay, so they were exposed to light. So the areas here, they were exposed to light. So I must make, let me go down a little bit on the diagram so you could see it properly. Okay, so these two areas were exposed to light. These were exposed to light. Okay, and the covered part here now, which is this part, let me quickly do that. So this part here, let me use a different color to represent that. Right, let's use red for this piece. So this area right here, that was covered, right? And so since it was covered, then there was no light in that. Okay, so this piece was covered. So this part represents the covered part. And since it's covered, then no light was penetrating that area. Now, what's supposed to expect? Because they're looking for the result of the student investigation. What is the result? This part was exposed. These two parts were exposed. This part was covered. No light was penetrating the leaf at that point, right? So if we use iodine on the entire leaf, then what you will notice here now is that the part that was exposed to light, it will photosynthesize. That part, those parts will photosynthesize and then produce starch. So the iodine at this point should change from a brown or yellowish brown into blue black. So iodine, let me just write that real quick. Iodine change from yellowish brown to blue black. Okay, to blue black. All right, so that would be the result. Okay, so the change from yellowish brown to blue black. So what do you see in these color in this area would be blue black. Okay, and of course you could put a color there as well to say, okay, this would be blue black. So if you want to shade that in as well, you can be able to, you know, put some shade in there and say that's blue black area and so on. Okay, just to make it look really nice. All right, but they didn't ask you to color it. Okay, just say draw and label on it. All right. Now in this portion that was not covered, let me just show you real quick what happened to that as well. Since there was no light, then there's no photosynthesis. And if there's no photosynthesis, then this part will be no change in color. So the iodine will remain, so the iodine remains yellowish brown, okay? Yellowish brown. So there's no change. So what color you'll see here is pretty much that yellowish brown color for iodine. So I'm gonna put it right here, okay? So it'll be like that type of color. So those are the color changes you'll see in the leaf, depending on if light is present or no light is present, right? So again, the shaded part, no photosynthesis, iodine will not change. The part that are exposed to light, those parts will change, all right? And you will see your blue-black color. You will see your blue-black color, okay? So just to note that. All right, so I promised you to show you the easy way to outline the test for starch. And so there's a 4-step method to test for starch. And it involves 4 things. It involves boiling with water. So you have to boil a leaf with water. So first is boil. So boiling first with water. Okay, so boil with H2O. Let me say water. Okay. The next thing you want to do after boiling with water is to boil with alcohol. So boil with alcohol. So boil with. So just in case you may need this, you may be able to come back on this. So boil it alcohol. And the next step you want to do is to soften with warm water. So soften with water. Okay, and that is warm water. Okay, let's put warm water there. Okay, with warm water. And then the last thing you want to do after this, that with the piece that comes after this, let me put it at the bottom here. The very next step, okay, that comes after this is, let me put it down, I'm missing that. Okay, so the very next step, the last thing, okay, will be this, test with iodine. Okay, test with iodine. So if they should ask you to explain or test a leaf for starch, these are the 4-step method you want to use. Boil it water, boil it alcohol, soften with warm water, and then you test with iodine. So those are the 4-step method how to test a leaf for starch. Okay. All right. So jumping over that. All right. There's some additional information there. Let's go to another question here. Question number 6. Okay. And question number 6 reads that 3 students were examined by a nurse. Their heart rates were recorded 3 times at rest and 3 times after doing 20 jumping jacks. The information is recorded in the table below. And if you look at this, you'll see the information. So let's just kind of analyze the information. You have a student here, F, G, and H. The heart rate at rest, and it's saying beats per minute. And so if you notice, there are trials, 1, 2, 3 trials. The numbers are there to see the amount for each trial. Heart rate after exercise, beats per minute. Again, we have 1, 2, 3 trials, and the numbers are there for each trial. And then at the end, now say average heart rate, beats per minute, and we have at rest. So all the students were the same heartbeat at rest. And after exercise, they gave us 2, which is student G and H. One is average 100, and you add up all these numbers here, divide by 3. That's all they did. And the same thing here, add up all these numbers, divide by 3, you get 90. So part A1, it said, why did all students do the same number of jumping jacks? And the same number of jumping jacks, the jumping jacks will be used as what they call a control. So it's a controlled variable, okay? So the number of jumping jacks will be a controlled variable, which means you expose them to the same conditions, okay? All right, so all the result will be based upon this. So you want to maintain accuracy, okay? You want to maintain accuracy. So the number of jumping jacks were used as a controlled variable, okay? Or because the jumping jacks were used as controlled variable. All right, so this is a controlled variable. All right, and the reason for that is to maintain accuracy. Why? Because you want to make sure all of them are doing the same thing, and none will have an advantage or disadvantage in any way. Okay, because of course if the variables are different, then of course the result will not be accurate in terms of comparison or comparing the difference between the thing. Okay, so it uses a control variable to maintain what? Accuracy. All right, so let's now move on to part 2. It said complete the table by calculating the average heart rate after exercise for student F. So let's go up here. After exercise, we have the values 80, 90, and 70. Okay, and so 80, 90, 70. Let's do the calculation on the bottom right here quick. So it'll be 80, 90, 70. So we're going to put 80 plus 90 plus 70, which will give us here 240. And so you say 240 now divide by 3. So 240 now divide by 3. And that now will give us our 80. And of course, the easy way to do it, if we move 10 from here to the 70, get all numbers being 80. That's an easy mathematical way as well. But do not try it unless you really understand it. So this one is 80 right here as well. Okay, 80 is the average. All right, that's an easy calculation. All right, so let's move on to the next part of the question here. And now I already go ahead and do this to save time. This one is said plot a bar chart to show the average heart rate after exercise for each student. And if you look at the average, again, one is 80, which is student F. Student G was 100, and student H is 90. And so you scale your axis and you label all the axis. And of course, you could put the wording here as well, because this is heart rate per minute. So you need to put that as well. Please label the graph, relabel the axis. At the bottom here will be students, okay? The student will be here, heart rate per minute will be on this Y. Matter of fact, let me put it. Okay, so please label your graphs always, okay, per minute. Please do not leave your graph blank without any labeling, or just put numbers there and just leave it, all right? And at the X will be student, okay? Student F, G, and H. Okay, great. All right, so let's now jump on to the next part of the question here. It's a state effect of exercise on the heart rate of the students. So what you want to notice here now, as heart rate increases, as exercise increases, then the heart rate also increases, right? So exercise causes an increase in the heart rate, okay? All right, so let me do this real quick. So caused an increase in the heart rate, okay? All right, just to make that real quick. All right, so that's what is happening here. So exercise caused an increase in the heart rate, or it caused the heart rate to increase, okay? Caused the heart rate to increase. All right, so that's part that for that. It said, the next part here said, use the result to state which student is the fittest. Give a reason for your choice. So if we go back to here, and this is after exercise, we notice F as the lowest heart rate, G the highest heart rate, H will in the middle. And so the fittest student will have the lowest heart rate, okay? If the heart rate is the lowest, that means that person is the fittest one. So this will be student F. So student F will be the fittest, and of course, G will be the least fit, okay? So student F. Now, but why is student F the most fittest? And because after exercise, so after exercise, all right, after exercise, the heart rate is the lowest, okay? Is the lowest, which indicates that the heart work less to pump the required blood to the cells, okay? Work less to pump the required blood to the cell, to the cells. All right, and when we say required blood to the cells, because remember the blood will go to the cells to provide oxygen and glucose for respiration. So because the heart is strong or fit, then it don't need to work as hard as the other heart or the other students, okay? So it's a fitter person do not need to pump hard. So therefore it could pump blood with a great pressure, with a great volume, with least amount of energy or exertion, okay? So you need to pump really less compared to the rest. All right, so part D now is a name one substance that would have a similar effect on the heart rate as exercise. So one substance, and this substance here is adrenaline. Remember, adrenaline is the flight and fright hormone, and so when you're excited, when you're scared, when you're frightened, and so on, this will chip in, and so it causes your heart rate to increase, it will cause the pupil to dilate, and so on, all right? And you produce a lot more energy from your glucose that rush to your cell, okay? So adrenaline is the answer. All right, so let's go to our next question, the question number 7, and we are almost out of here. All right, so question number 7 is said now, this question is about pollution and the problems that it can cause. Explain what is meant by the term pesticide. So when we talk about pesticides, so pesticides are chemicals, okay? So a pesticide is a chemical used to control pest, okay? So a pesticide, let's put it right here. So a chemical used to control or even destroy, to control or destroy pests. Okay, so you're going to destroy them. Okay, all right. And just to point out something right here. Pesticides are categorized in different categories. So let's just make a note of that for your sake. The types of pesticides, or the examples of pesticides that I may put here, we have those that are called fungicide. So fungicides, they will kill fungus. Okay. We also have herbicides. Herbicides, they will kill plants or unwanted plants. And we also have what we call insecticides. Okay. So insecticides, they will kill insects. So those are examples of pesticides that are used especially by farmers. Okay. All right, so now let's jump into part B. So the diagram shows how pesticide is in runoff water built up along a food chain in a freshwater ecosystem. All right, so the pesticide will build up, and so we call pesticides that will remain in organism persistent chemicals or persistent pesticides. All right, so here we have the plankton, and notice the amount of pesticide in the plankton, not so much. The fish will have more, all right, and the grebe will have much more than the fish and the plankton. Okay, so now let's talk about this. Okay, and so here now it says state the term used to describe the build-up of chemicals along a food chain as shown in the diagram. So as chemicals become more concentrated or built up along the food chain, the process that is called there is called bioaccumulation. Okay, because it accumulates in living things, right? So bio, living, accumulate means to build up. So it's called bioaccumulation. Okay, accumulation. The bioaccumulation. Okay, all right. And so that's bioaccumulation there. And now it said, referring to the named organism in the food chain, explain how this process occurs. So we're going to use the names to talk about how bioaccumulation takes place. So I want to notice something here. Starting from the lower level of the food chain going up, the chemical is increasing. But why is this so? And just to kind of generally explain to you real quick, is that as I go along the food chain, energy decreases, yeah? And so as energy decreases, then the organism eat much more, and so therefore the poison or the chemical will build up. Okay, so let me just do this real quick. And so let's start with the plankton. So the plankton, okay, so the plankton takes in the pesticide. Okay, the plankton takes in the pesticide. Okay, that's the first thing happen. Okay, the plankton takes in the pesticide, and now since energy decreases along the food chain, okay, the organisms will eat more. All right, we'll eat more of the previous organism, okay, of the previous organism. And why will it eat more of the previous organism is simply to fulfill its energy requirement. Okay, it's a simple to fulfill its energy requirement. Okay, all right, very important for us to know that. All right, so we're not finished here yet. So again, I'm going to add some more to this. Okay, so the plankton takes in the pesticide. Since energy decreases along the food chain, the organism will eat more of the previous organism. So therefore, the grebe, let's start with the fish. The fish will eat a lot of the plankton. Okay, so the fish will eat a lot of the plankton. Okay, so that's very important. The fish eat a lot of the plankton. All right, and the grebe now will eat more than one of the fish. Okay, and so because of this reason now, because of eating more and more, and the chemical is not decreasing, the chemical is actually building up. Okay, So the more you eat, the more the chemical built up, okay? And remember now, the larger organism will eat the smaller one in larger quantities. And so eat them in large quantities, then will cause the toxin or the chemical to build up. Okay. All right, so part C of this question here now, it said that the graph shows the result of a study carried out on the thickness of bird eggshells in areas where pesticides containing DDT were used. Okay, and DDT is now a banned substance, right? In almost, well, in most places, DDT is now a banned substance. All right, and so you have a thickness of eggshell on the Y and the concentration of DDT on the X, and this is per dry mass of shell, okay? And so if you notice here, as the DDT increases, notice as the concentration of DDT increases, now what is happening to the eggshell is becoming less. So less concentration, thicker eggshell. More concentration, thinner eggshell, okay? All right, so that's very important for us to note that. It's a use a graph to describe the relationship between the concentration of DDT in the eggshells and the thickness of the eggshell. So here is saying now, as the concentration of DDT increases, then that of the eggshell will decrease, okay? So as the concentration of concentration of DDT increases, the thickness of the eggshell decrease. Okay? All right. So that's very important there. So as one going up, as the concentration of DDT increases, then the thickness of the eggshell will what? Of the eggshells will decrease. Okay? All right, great. Now, the next part here says use a graph to suggest what effect high levels of DDT might have on birds population, on the bird population. Explain the answer. Now, as we established in the previous part of the question, if concentration of DDT increases, the eggshell, the thickness of the eggshell decrease, right? Remember, the eggshell is used for protection and also to maintain the embryo as it develops, correct? All right, so maintain that suitable environment for hatching to take place. So if the concentration of DDT increases, then the population will decrease. So you'll have here a decrease in the population. Okay, so that's what you'll have there, a decrease in the population. All right, so that's what you'll have. And so the population, and the reason for this is because higher concentration or higher DDT concentration, okay, so higher concentration of DDT results in thinner eggshells. All right, hence lower probability or lower possibility, hence lower possibility or chances, lower chances of eggs being hatched. Okay, so that's very important. So lower chance of eggs being hatched, because the thinner the shells mean that the embryo will be susceptible to different conditions such as temperature. So the temperature could also denature the enzymes in the egg. All right, you know, if the egg is too warm, then it will spoil, or if moisture get into the egg again, it could also spoil. All right, and so those are very important things. And even if certain gases get inside of the egg, it could also damage the egg as well. All right, so the last part of this question here said the birds are predators of snake. Okay, so the snake is the prey and the birds are predators. Just to explain how the effects or the effect described in C2 would impact the snake population. So this will happen now. If the birds are eating snake, right, and the population of birds decrease, then if you have a decrease in birds that eating the snake, then the snake population will what? Increase. Okay, so the population here will increase. Okay, the population will increase. So the population of the snake will definitely increase. Okay, we have that right there. Let me go back here. I think I made a mistake here. I put pollution here. Decrease in pollution. Decrease in population. I don't know why I made this mistake. Okay, so let's quickly correct this one real quick. So here is a decrease in population. Okay, the decrease in population of the bird. So decreased population of the bird, and so you have an increase in population of the snake. So the population of snake, let me get into this, let me do this properly. So here you have a population of snake will increase. Okay. All right, good. So now we have everything correct. All right, great. All right, so again, now let's jump on to another part of the question here. And this is a 2 mark. Okay, so the population of snake will increase, and why it will increase? Because the population of bird will decrease. So you have less predator. Okay, and so the population of bird will increase, and this is because you have less birds eating the snake. Okay, so that's the reason why. So the population of snake will increase, that's a mark, and then less birds eating the snake because of a decreased population. All right, great. All right, so that's your 2 marks right there. Okay. All right, so let's go to now our final question. It's a diagram shows the simplified chemical structure of a portion of a chromosome found in a cell nucleus. And so here now we have box D, we have some letters here, we have P, we have Q, we have R. Let's figure out what is happening here. First and foremost, is a state the name of the molecule shown in the diagram which chromosomes are made of. So this chromosome are made up of this structure, and this is called our DNA. Okay, so this is DNA. All right, and so the structure of DNA, we have a number of things. I'll point this out real quick for you. All right, and so that's our DNA. All right, so the answer here is supposed to be DNA. State the name of this, which is DNA. I'm going to write out the name. So DNA is just the abbreviation. So DNA is deoxyribonucleic acid. So deoxyribonucleic acid. Okay, so deoxyribonucleic acid, that's the name. All right, and it said in part B here now, it said identify the parts labeled P, Q, and R. All right. So P here now is what they call a deoxyribose sugar. So D is a deoxyribose sugar, or what they call a 5-carbon sugar. So D is a deoxyribose sugar. Let me just put it a little bit up here. So it's called a deoxyribose sugar, or otherwise called a 5-carbon sugar. So it's a 5-carbon sugar. Okay, 5-carbon sugar. All right. So in other words, a pentose, right? So it's a deoxyribose sugar, or a 5-carbon sugar. For Q, it is what they call a nitrogen base. I'm going to give it a specific name right here. All right. So it is a nitrogen base. All right. So let's type that in real quick. All right, wrong part. All right, so here now we have nitrogen base. All right, and the exact name for this, I'm going to give it, is called cytosine. Okay, so it's called cytosine. All right, and cytosine is what is represented by a C. Okay, so this be C, that will be there, so it's called cytosine. Okay, so it's called cytosine. All right, and so again, it's symbolized by C. Okay. All right, so let's put that on the diagram. Let's do that real quick. All right. Let's see something here. Let's go up. Cytosine, the C right here. All right, so I'm gonna put all of them for you. I should put R first, so that's C right there. I'm gonna tell you the others as well. And R, which is the phosphate group. So this is the phosphate group here. We go all the way back. All right, it's a phosphate group. This is the phosphate group. All right, so the phosphate group. So there are three things that make up the DNA. You have a 5-carbon sugar. All right, so you have a 5-carbon sugar, which is called deoxyribose sugar. We have a nitrogen basis called the cytosine. So let me just correct this. It's actually, come on, it's actually S right here, right? So cytosine. All right, and a phosphate group. So those are the three parts that make up a DNA structure, right? And then now let me just show you this real quick. In the DNA, T is always bound to A, okay? So it's thiamine and adenine, okay? So thiamine and adenine. And so wherever you see a T, you'll see a A. So A is right here as well. A T is here because there's an A on the opposite side. Wherever you see G, you will see T. So always remember that T, which is thiamine, always bound to adenine, so of A. And guanine, which is G, always bound to C. Okay, always bound to C. All right, so once you cover that, all right, so next part here now is to place the correct missing letters in box D, which we did, right, which was A. Let's go back up there. So this is A. Excellent, right, so box D is A. All right, so let's jump down to the next part here. All right, it said, before a cell divides by mitosis, the molecules shown in the diagram must be carefully replicated. Explain why. So explain why replication of the DNA must take place, or replication of even the chromosomes must take place. And this is, well, the basic reason, or the primary reason here, is to ensure that the daughter cells, and so the daughter cells, now the daughter cells, to ensure that the daughter cells have the same number of chromosome or DNA, okay? Same number of chromosome, okay? All right, so same number of chromosomes or DNA, or at least the daughter cells have the same genetic information as the original cell. Because this is mitosis. Mitosis produces cells that are genetically identical to the parent cell. And so the genetic information, just to put this here again, just additional information here. So the genetic information in the daughter cells, will be the same as the parent cell, okay? So all the genetic information will maintain. So maintain the number of chromosome and also maintain the genetic information, right? So the cell will be identical and have the same genetic makeup, right? That's what mitosis is for, produce the same type of cells, okay? All right, so this part of the question is said that the diagram shows 5 different stages of mitosis in a cell. And so we have 1, 2, 3, 4, and 5. And now this asks us now to identify, it said list the diagrams. So list the diagrams 1 to 5 in the correct order to show the stages in which mitosis occur. And so let me just quickly label these for you. Based on the structure, let's start with number 1. So number 1, notice the chromatids are going oppositely. So this is what they call the anaphase. Okay, so this is anaphase. All right, and here now, for number 2, you can see that the chromosomes are coming together. The nucleus is the nucleolus is not there as in this one. So this one here, which we call the prophase. Okay, this is the prophase. All right, and then this one here now is called the interphase. This will be the first one when you only see those thread-like structures. All right, and so the chromosome occur like a thread-like structures. All right, so this is the interphase, and this one here, that the chromatids are in total opposite places, the cell ready to divide, and this is called, we call it telophase. Okay, so that's the telophase. And the chromosomes are lying in the middle here, so this is called the metaphase. Okay, and so remember is PMAT, IPMAT, and then after the telophase you have what they call cytokinesis. So it's something for you to always remember. Let me just put it right here real quick, right? Is this you need to remember when for mitosis, you have the interphase first, and then you have, after interphase, you go to PMAT, okay, which is prophase, metaphase, anaphase, telophase, and after the telophase, you have what they call cytokinesis. So you got a C right there, right? So just to make a note of this, that's the order in which cell division is taking place. All right, very important. All right, great. So let's move on to the next part of the question here now. It asks us to identify these in order. So in order, again, the first thing, we say in order, in the correct order. And so the first thing that will take place here, the very first part, as I mentioned, I, P, MAT, C. So the interphase will be first. So we're going to have interphase first. All right, so interphase will be the first thing up in here. So let's put it here, number 3. Okay, so 3 will go into, so after 3, it will go into, let's see what come after interphase is prophase. Prophase, what number again? Number 2. Okay, so number 2. Okay, I could remember the rest now. So number 2, and then that will go into number 5, which is the metaphase. So after P, then metaphase. So that's number 5 there. And then after that, we will jump into number 1, which is the anaphase. That's number 1 there. And then I'll jump into the last phase here, which is number 4, which is the telophase. There's no cytokinesis showing right here now. So after 1, then we go into 4. Okay, so that's the order: 3, 2, 5, 1, and 4. Okay, great. Now we say that state which diagram presents anaphase. Give a reason for your answer. So again, we established that diagram 1 with our anaphase. So diagram 1 is the anaphase, and so let's just quickly tell you the reason why. And this is because the sister chromatids, all right, are pulled, or they can either say they are pulled or moved, whichever one. They are pulled to opposite poles, all right, of the cell. All right, and this is done by the spindle fiber. Okay, so just to make a note of that, it's caused by the spindle fiber. All right, so this will now be our answer right here. Okay, all right, so great. So now we are finished with this paper. It's the end of another review, and please just take your time, go through it, make sure you understand the questions, understand the answers as well. And this will always say take care for now. I'll see you soon for some paper 3s, and I may do another paper 2 before go to paper 3s, but we will be doing some paper 3s definitely. So see you next time. All right, take care and be safe now.